Good morning, all of you. Today, I'm going to share my work to describe the impact of bird environment and behavior on eggshell cuticle quality and food safety of table eggs. The Canadian egg industry produced approximately 10 billion eggs in 2009 with a value of about $1.56 billion. Avian egg, as we all know, is a natural containment for developing embryo. Moreover, table eggs are amongst most nutritious and economical source of protein for human consumption. Eggshell cuticle plays a very important role in maintaining food safety of table eggs. It's first line of defense against invading pathogen, and it functions as both physical barrier as well as antimicrobial proteins present in the cuticle act as chemical barrier in order to reduce contamination by pathogens. Outer surface of eggshell cuticle has a patchy distribution with cracks and fissures, which could affect or impact its interaction with bacterial pathogens. In the United States and Canada, eggs are washed before the retail sale in order to remove potential pathogens. And despite of this practice, major gram-negative or gram-positive pathogens are introduced into the food chain through contaminated eggs. And this results in food safety-associated recalls, which is a major public health concern. And in addition to economic losses to poultry industry, this results in negative publicity as well as reduces consumer confidence in nutritious egg. Therefore, it is very important to maintain food safety of table eggs. In our previous study, we have shown that cuticle quality can be affected by hen age, hen strain, egg washing, and there is literature on effect of hen health on cuticle quality. However, there is limited information on nesting behavior or housing system and how it affects cuticle quality and thereby food safety of table eggs. In previous study, it was shown that eggs with good quality cuticle completely prevented E. coli penetration as compared to eggs with poor quality cuticle, where E. coli could easily penetrate eggshell and contaminate egg contents. Moreover, in our previous study, we have shown the adherence of salmonella cells on eggshell cuticle and shown the effect of egg washing. With respect to egg washing. And here we quantified both cuticle indicated by red and bacterial indicated by green fluorescence using confocal microscopy. And we observed that in ungraded eggs with good quality cuticle, where the cuticle was higher, bacterial adherence was lower, and when this was removed by bleach treatment, bacterial adherence was high, and overall quantification showed a trend of higher bacterial load on bleach-treated eggs as compared to ungraded eggs. Therefore, in this study, we evaluated cuticle quality and chemical composition on a shell surface of eggs from different housing systems using Fourier transformation infrared spectrometry, and we quantified adherence of bacteria on eggshell surface from eggs from different housing systems using agar plate count method. So here are the results. Bacterial load on no cuticle, that is bleach-treated eggshell surface, was significantly higher as compared to eggs from different housing system. Moreover, a closer look at the data showed that bacterial load and on the surface of eggs from free-range eggs was significantly lower as compared to enriched. Next, we evaluated chemical composition using FTIR, where we observed a strong negative correlation between total cuticle and carbonates, a positive correlation between sulfates and carbonates, and a positive correlation between total cuticle and sugars. Based on positive correlation between total cuticle and sugars and phosphate, we can predict that cuticle has a compositional gradient with which could be divided into two layers, inner and outer cuticle, with different antimicrobial proteins in each layer. We observed differences in cuticle chemical composition within housing system, where total cuticle was higher in enriched and free range as compared to free run, and sugars were higher in enriched as compared to free run. Conversely, sulfates were higher in free run as compared to enriched, and carbonates were higher in free run as compared to enriched. So overall, we did not observe large differences in salmonella adherence between the caging system. However, trends showed some significant difference with lower bacterial load in free range as compared to enriched. Chemical composition was measured using FTIR, and total cuticle was higher in enriched and free-range eggs as compared to free-run. Overall, these results indicate that cuticle quality and therefore food safety can be used as index of bird welfare. We have designed follow-up studies in order to determine reproducibility of these results and also to quantify variability in housing systems at farm levels. Overall, our results would provide new tools for predictive microbiology to improve food safety of table eggs, and also identification of anti-adhesive cuticle components can be used for various applications to prevent bacterial adhesion on medical devices, coatings, or food production equipment. We would like to thank our collaborator, Professor Alejandro from University of Granada, for FTIR measurements, and also we are thankful to Bunbury Farms for in-kind support and our funding support from EFC, HELRIC, and EFO. With this, I would like to thank you all, and I look forward to your questions. Thank you.
Well, thank you very much for that great presentation. And again, I would like to apologize if I mispronounce your name.
That's all right. It was perfect.
So I see we do have questions here from Vincent Guillonnet. Could you expand on your comment that the cuticle and food safety is a reflection of animal welfare?
I would say that right now the study is ongoing study. So of course, I cannot completely confirm that there is a connection between cuticle and animal welfare, because in this study, I evaluated eggs from one farm. So in my follow-up study, I would like to evaluate eggs from different farms to see what is the variability at farm level, and thereby I would like to comment on this aspect of cuticle and animal welfare. But overall, I believe that there is different level of stress in different housing systems, and this stress could affect cuticle deposition, because there are previous studies which have shown that stress from pen to cage transfer has an impact on cuticle deposition, and birds are more calmer in curtain nest as compared to other housing systems. So this stress level could affect cuticle deposition, and this could be a reason to correlate it with animal welfare.
Thank you. And I think that somehow answers the following questions by Tina Widusky to discuss the mechanism by which housing system would affect the cuticle. So I would take it from your answer, it's beyond the impact of housing on stress. It's like,
yeah.
Now, I've got a follow-up questions on that, and that's fair enough, that the stress may be a factor, but did you control for the diet of those birds, especially when they are in free range or outside and they have access to uncontrolled source of ingredients?
Yes, I totally agree with you on that aspect. Like, we received eggs from Bunbury Farms, so we were not directly involved in rearing of birds. So I hope they controlled dietetic aspect of this study. But yes, I can follow up with Bunbury Farms to look at what were specific diets in different housing systems.